Hi everyone. The title of our paper is Uni-Hema, Unified Model for Hematopathology. My name is Abdul Rehman and I am a PhD student and working under the supervision of Dr. Mohsen Ali and Dr. Waqas Sultani. And we are from the Intelligent Machine Lab at the Information Technology University. In all over the world, the conducting percentage of the blood-related test approximately 55%. And from that 55%, almost 42% are blood smear slide analyzed further in microscopic. This is a complete field of view of a blood smear under the microscope. And in blood, we have a three types of a hematological components. The first one is the white blood cells, second one is the red blood cells, and the third one is the platelet. Medically, we can divide the blood test into two categories, hematological disorder and infectious diseases. In the hematological disorder, it is the change of the morphology of the base component, and in the infectious diseases belongs to the new parasites or appear in the blood components like ACS and the OCS families. In the hematology, we have a different types of a task that belongs to the diagnosis of the diseases. The first one is the cell detection. The cell detection is basically the identification and localization of the abnormal cells in the complete field of view image. The second one is the morphological prediction. The prediction of the morphological feature indicates the prognosis of the patient. Third one is the segmentation. Basically, the segmentation of the abnormal cells shows how much precisely change occur in the cell shape. The next is the cell classification. The cell classification mostly important in the bone marrow, where the cell type is changed with respect to the minor changes of the morphology. Lastly, the question answer and the mass language model. Task for the detailed conversation with respect to the complete field of view and the single cell. So in hematopathology, we have a different types of a challenges. The first one is we have a multiple diseases that can be diagnosed by the blood samples. Secondly, we have a complete field of view images that capture the multiple cells and its context for detecting the abnormalities. And further, we have a single cell images that allow the detailed classification of morphology of the individual cell. Third, we have a multimodality data like the text data belongs to the mass language modeling and question answering and with respect to the images. So for the hematopathology, we propose a solution that can perform the multiple tasks like single cell classification, cell segmentation, morphological prediction, abnormal cell detection, visually mass language model, and visually question answering. And we call that unified model as a Uni-Hema. All the dataset we use in this research is publicly available. For the detection task, we gather the 17 publicly available datasets that belongs to the different diseases, and after combining them, we have a 13 different classes of cells. For the cell segmentation, we use the 11 dataset from the public resources and most of them are binary. So we converted the non-binary samples into the binary. Most of the largest dataset are belong to the single cell images from the blood and the bone marrow. So we use the 16 dataset and by combining them in the result, we have a 45 class. So for the multimodality dataset, we use the two morphologically aware dataset and created the VN mass language modeling and VN question answering dataset. Totally, we have a 46 dataset containing almost 0.7 million images. Is the overview of model architecture. We have a VN stream based on the ResNet 50 backbone and visual encoder and decoder. Secondly, we have a text stream based on the T5 encoder and decoder based model. And in the proposed multi-modal HEMA former, we use the deep features from the multiple hierarchical level to perform the different tasks. For the single cell classification, we use the R embedding of the encoder output, and after pooling them, we concatenate a learnable query Q1 and then use a classification head to classify the cell type. In our corpus, we have a different types of a diseases. So during the training, we use a disease-informed prompt corresponding to the each input image to distinguish the disease and added a text-guided refinement module to attention the top K refined embedding with respect to the disease before passing the decoder to perform the object detection and morphological prediction. For the cell segmentation, including the text-guided refinement, we incorporate the initial spatial layer of the backbone and its corresponding embedding after the encoder. By adding them, we project them and use the object-enriched decoder output embeddings by using the Einstein summation to generate the binary mask. After that, we use the upsampler to resize the mask with respect to the input sizes. Finally, for the VN question answering and VN mask language modeling task, we use the backbone embedding and the text encoder embedding. After projecting the text embedding, we concatenate the learnable queries and perform the attention fusion between the text and the image embedding. Then we project the embedding with respect to the text decoder and pass to the text decoder for the complete sentence generation and the answer generation. In Uni-Hema, we have 4 different modules to learn the different level of hierarchical information to perform the different tasks. These are our results with comparison to the SOTA 1-to-1 method. These are the These are the results of the Uni-Hema with comparison to the state-of-the-art one-to-one method and multitasking method. The performance of the unified model is better or comparable with respect to the different tasks and with respect to the different data sets. So these are the results of the other domain shift dataset to show the generalization of our method. Similarly, against the foundation model, the performance of the Uni-Hema is better against the hematological task and with respect to the different datasets. This is a TSNE plot of the foundation model and the Uni-Hema feature of the two dataset display clearly class separation is much better of the Uni-Hema. These are the segmentation results and comparison with the state-of-the-art method and our method on the anemia, malaria, and the white blood cell images datasets. The, the true positive, and false negatives are shown in the light yellow, blue, and the red with respect. These are the segmentation results. These, these are the segmentation results comparison with the state-of-the-art methods on anemia, malaria, and WBC dataset images. The light yellow, these are the segmentation results comparison with the state-of-the-art method on anemia, malaria, and WBC dataset images. The light yellow color shows the true positive, blue color shows the false positive, and the red color shows the false negative. And our method reduced the false detection and improved the localization, especially for anemia and the white blood cell dataset images, while handling malaria robustly. These are the some examples of the visual question answering, and that shows that our method can generate approximately the correct description with respect to the image and the question. These are the qualitative results of the detection. The dotted white boxes denote the misdetection, while the dotted color boxes represent the incorrect classes prediction. Across the diverse dataset, our single unified model achieves strong localization and class consistency, performing our matching the specialized detectors in challenging cases. Thank you so much. The link of the GitHub repository and the email of the corresponding author are available in the description. Thank you again.